Hello, this is Jeff Stuckey from Bruker Fluorescence Microscopy. I'd like you to welcome to our webinar today, in vivo imaging of the tumor microenvironment of breast cancer. The presenter today is Joe Chilsevsky. Joe's a graduate research assistant in the lab of Patty Keeley at the University of Wisconsin. The major focus of their research there is the microenvironment and microarchitecture of breast cancer tumors. And you see from the title, this is going to be the main focus of his talk today, using multi-photon imaging modalities in order to study the microenvironment of breast cancer tumors. When we get done with the presentation, if you'd be so kind as to take a couple of minutes and fill out a brief survey that'll be at the end, much appreciate that, so we can monitor the quality and do what we can to improve our webinars. The other thing to keep in mind is ask any questions. There's a question section on the interface of the webinar. Make notes of any questions you have, and we'll take all questions at the end of the presentation. So with no further ado, I'd like to welcome Joe and tell us about the research they've been doing there. Joe?
Yeah, thank you, Jeff. And I'd like to thank Jeff and Bruker for this opportunity to talk about my science today. As Jeff said, I'm a fifth-year graduate student at the University of Wisconsin in Madison, and this is a picture of our downtown area. And this work is also in collaboration with a on-campus affiliate, the Laboratory of Optical and Computational Instrumentation, that is headed by Kevin Elisari, and my advisor is Patty Keeley. And I'm going to be talking to you guys today about my work working with multi-photon imaging, looking at in vivo imaging of the tumor microenvironment in breast cancer. So I'd like to start off these talks with this image. Kind of sums up a lot of what we do. What you're observing here is the tumor microenvironment, and what you can see here is the blue cells are actually the breast tumor in a live animal model, and the red cells are components of the tumor stroma area, and then we also have this green collagen fibers, and this is what really makes up a lot of the tumor microenvironment. But what's really compelling about this image is I'm looking at a live animal, but I'm using no artificial fluorophores or dyes, exogenous fluorophores or dyes. This is all endogenous fluorescence that we can get for free. And so what I'm going to talk to you about first is what our lab does. A lot of background about collagen alignment. So we're going to talk about the green part of this image. But then I'm going to follow up with the findings of my paper looking at the blue tumor cells and identifying the red cells, where I hope to kind of show you today that these are we believe to be infiltrating macrophages inside of the tumor itself. So our lab really was inspired by obviously research that happened before us, but one of the major observations was this Norman Boyd paper where he showed that mammographic density can be associated with breast cancer development and risk. So what I'm showing you here are different classifications of different breast mammograms. And you can tell that they're different as they go increase in opacity. So they increase in their breast density. And what he found in his 1995 paper is that just by correlating the density of these mammograms, he could then correlate to the relative risk of development of breast cancer in that patient. So that suggests that something that's causing this mammographic contrast is in and of itself might be a precursor or a prominent risk factor for the development of the disease. And so there was a paper that came out a little bit afterwards by Alawami and everyone, also from Boyd's group as well, that try to characterize what are the major players that are causing these increases in density in mammograms. And what they found was that when they looked at histological stained sections from breasts with high mammographic density, they had a significant increase in their collagen deposition compared to mammographic densities in low. This strongly suggests that just increasing collagen alone might be playing a very critical role in the development of breast cancer. So our lab published a series of papers in 2009 to 2011 that suggested that collagen has a direct link to breast cancer in vitro and in animal models. And so we'd like to highlight a couple of these really elegant experiments that our lab did before my time. First one is we took normal breast epithelial cells in vitro, these MCF10As, and we put them in a three-dimensional collagen matrix. And all we did was we changed the amount of collagen from low density and high density, and we significantly changed the phenotype of the cells. So by increasing, in low density, we have this normal breast ductal architecture, but by increasing collagen alone, we seem to switch the phenotype of the cells into this very invasive group, suggesting that collagen is indeed changing the phenotype of the cell into a more advanced and aggressive form of the towards cancer. What we also did is we studied this in a mouse animal model, and so we had the ability to change collagen density in an animal model by using this mutation inside of the animal itself using a COL1A1. This mutates the collagen so it's less likely to be degraded. And this gave us an animal that had an increased amount of collagen deposition. So we had a high density animal model and a normal amount of collagen density model. So we manipulated that and just crossed this with our already well characterized cancer animal and found that cancer progression sped up. We had an increased amount of tumor volume over time, but more interestingly, we saw an increase in the amount of lung metastases, suggesting that not only are we increasing the development and speed of the cancer itself, but we're creating a more aggressive disease. So this suggests that collagen alone is linked to the progression of breast cancer. So in order to study this, we utilized a nonlinear optical technique called second harmonic generation with multiphoton microscopy. And to kind of go over a little bit of what second harmonic generation is, it's a physical phenomenon that when you use a laser light source and you shine it through a medium that has these two major characteristics, it's highly polarizable with a large dipole moment and has non-central symmetry, what you get is a second harmonic wave that is exactly half the incident wavelength. Lucky for us, collagen in its macrostructure and fiber has these two characteristics of being highly polarizable and non-centrosymmetric. So we can actually generate images of collagen fibers non-invasively without any labels or dyes and actually look at the collagen architecture. There's a couple sources here, especially this protocol paper from Paul Campagnola's lab in Nature Protocols that go over more detail about how that process works. So we use this technique to really ask a really basic question. How does collagen architecture look in normal mammary gland? And so we took an excised images of our mammary tissues from mouse and rat. And we find in normal tissue, collagen architecture looks, how I kind of describe it as kind of a bowl of spaghetti. We see really squiggled fiber structures. There's not a lot of influence. And in the middle here would be a normal breast duct. And so what we did is we used second harmonic generation and excised tumors at different time points in our classification of our tumor, our mouse cancer model at different time points that correlate with different stages of the disease. And we found and characterized these different tumor-associated collagen signatures. So at a really early stage where we would expect the initial progression of the disease, we see something very different already from normal stringy collagen. We see abnormal deposition of collagen around the developing mass. And we refer to that as a tumor-associated collagen signature 1. As the tumor grows, we see something even more interesting. We see this abnormal straightening of the fibers around the developing benign mass. And they seem to align themselves parallel to the tumor and the stroma boundary. And we refer to this as TAX2. But more interestingly enough, when we get to the later stages of the disease where we expect to see circulating tumor cells and the development of metastases, so the cancer has progressed out of the tumor proper, we see this tight collagen fibers that were before aligned parallel to the tumor stroma boundary have now realigned themselves perpendicular to the tumor stroma boundary. We refer to that as TAX3, or a very invasive phenotype. And so our lab has this model that as cancer is progressing and growing, we can actually use this as a biomarker for us to predict and or study how collagen interacts with the tumor microenvironment. And we think that TAX3 might be a super highway for cells to move away from the tumor into the stroma itself. And so here's an image of cells that are invading out along these straightened collagen fibers. And so this is all great. We did a lot of the study in mouse tissue, but is this clinically relevant? Do we see this in human disease itself? And so what we did is a scientist in our lab, Matt Conklin, in 2011, took what's called a tumor microarray, where he looked at 196 paired patient biopsies and found, and just asked the question, does TAX3 exist in these tumor biopsies or not, and put them into 2 classification groups using second harmonic generation. And what he found was that it does. So by looking at these paired patient biopsies, we have a significant amount of data that's paired with each one of them. And by correlating, putting the patients in the TAX3 category, we found that patients that had TAX3 had a significant decrease in the amount of time they had without the disease-free. So they had a decrease in the amount of disease-free, as well as they had a decrease in their relative, they had a decrease in or increased amount of death. So TAX3 was associated with a poor patient outcome, and there was an increased amount of the relative risk of relapse from 4 to 7-fold, just from having this second harmonic generation TAX3 signature alone. So that was really interesting to us. So one of the things that our group has been really working on is how to automate this process. So what we did is we took these images and we literally just drew with a protractor and a ruler the angles of alignment for the from the tumor to the stroma boundary itself. But with our collaborators at LOCI, Laboratory Optical and Computational Instrumentation, we were able to develop software technology to automate this process. And so what we did is that group there used what's referred to as a curvelet transform, which allowed us to look at each individual fiber in angle and create a vector. And so we can take an image like this and create a series of different vectors with different angles themselves, and then create a histogram of these angles. And then we can correlate that to a drawn tumor stroma boundary and see the relative changes of these angles of alignment. And what we found was that we saw that using this computer algorithm, we were able to see the same thing with that we observed with using it by hand with human measurements. And so If you're interested, if this is something that might help your work, we just wanted to kind of plug this data. It's open source, and it's located at LOSI, on their LOSI website, and it's CT Fire and Curvaline, and it's a really powerful process of being able to convert these fibers into different relative alignments. So I've already talked now about how the green part of this image is significant, and it's the background of our paper. But now I want to change gears into what are these blue and red cells, and how did we create these images? And what we're actually looking at is endogenous fluorescence of two metabolic cofactors, NADH and FAD. And so NADH and FAD are actually metabolic cofactors, as I said. And so this is a very brief description of basic cellular metabolism of a simple sugar of glucose. And you may remember from your basic biochemistry, or if you don't, the whole goal is to take a sugar and develop it into the energy unit of the cell, which is adenosine triphosphate, or ATP. And it does this by converting glucose into pyruvate through a process called glycolysis. And then pyruvate is shuttled into the mitochondria, where it enters the citric acid cycle, and where it reduces these metabolic cofactors of NAD and FAD into NADH and FADH2 that enter into the electron transport chain, which very efficiently converts or creates a series of ATP particles, or molecules, excuse me. So these are major players in metabolism, and they have different roles if they're reduced or oxidized. Lucky for us, these are also fluorescent, but with a very interesting fluorescent characteristic. NADH in its reduced form is fluorescent, but not in its oxidized NAD+. Conversely, FAD in its oxidized form is fluorescent, but not in its reduced form of FADH2. So this kind of gives us what we refer to as a metabolic snapshot of the cell, just by looking at these relative intensities alone. So a lot of our work, we use a Ultima IV multi-photon system, so this is actually a picture of our actual rig that we have, and we use this Zeiss 20x water immersion lens and a standard Insight laser for this research. But in order to look in vivo, where we can study the metabolism changes, we needed to develop a optical window into the tumor itself. And so with our collaborators at Albert Einstein College of Medicine in New York, the Candelas lab, we developed something called a mammary imaging window. And that's essentially a contact lens that we can surgically implant over a developing tumor mass in a live animal model. And what this does is it holds a cover slip right over the developing tumor itself, which allows us to then optically penetrate into the tumor environment. And so by hooking this up to our Ultima IV and setting a wavelength for excitation and our filters for NADH, we see a really beautiful image of our tumor architecture. So what we're looking at here is a large tumor, and these little black circles would be nuclei that's making up the whole mass of this particular tumor. But more interestingly, when we change the wavelength and we change the filter settings so that now we are exciting FAD exclusively and we are looking at FAD, we see something different. We see a whole new subset of cells that are inside of the tumor microenvironment, which is interesting. And so when we also then combine that with second harmonic generation that we can also do in vivo, and we combine this together, we're getting a snapshot of the metabolic properties of the tumor microenvironment. And we have two different subsets of cells without the use of an exogenous fluorophores or dyes. So it's all endogenous fluorescence, which is really interesting. So a really obvious question that we had was, what are these FAD high cells? The approach that we came up with in order to study this is by using in situ antibody staining underneath our imaging window. And so what that means is we developed something called a ported mammary imaging window, which is essentially what we did is we took a 30 gauge needle and we fed it through the side of the mammary imaging window, which gave us a port underneath that allows us to now introduce different dyes or stains, and in this case, primary conjugated antibodies, in order to look at the environment. And so this is another view, and here I took a montage, and what we're looking at here is underneath the window with the blue being second harmonic. We actually stitched many images together, and we have, this is actually the needle itself, and you can see the bore here and the tip of the needle here. So we're actually inside and manipulating inside of the tumor microenvironment area itself. And so what we did is we used primary conjugated antibodies that will allow us to specifically label cells and try to decipher what are these cells. And so here's an image. What we're looking at here is one field of view, and here's the bore of the needle, and this would be the thickness of the needle. The tip would be way off here, and we can see these bright FAD cells. And by using brilliant violet dye, we were able to spectrally separate out and locally inject temporally and spatially where we wanted to look at underneath the tumor microenvironment of these areas. And so by using brilliant violet 421 as well, which is a blue dye, we were able to spectrally separate out FAD high cells from this dye itself, so we could ask the question, where are these co-localizing? And so the first antibody that we used was CD45, which is a broad immunogenic antibody that labels all immune cells. And what we found was by injecting this with brilliant violet, we could actually see co-localization. So here's an image of a cell that is bright in FAD and bright in CD45, and they're co-localized. But we also saw cells that were not related to these FAD high cells at all. For instance, this cell is not bright in FAD at all, yet is clearly labeled by CD45. And so this suggests that the bright FAD cells are immunogenic, but there's a specific subset of cells inside of the tumor that are FAD high cells. So our hypothesis was that these are actually macrophages. So we took a macrophage specific marker of F480 and CD68, and what we found was that we saw a significant increase in the co-localization of FAD high along with the CD68 and the macrophage specific markers. This suggests that these FAD high cells are indeed macrophages of origin, and that what we're looking at is specifically macrophage, or at least it suggests that it is. And so another thing that we can do and tease off with endogenous fluorescence is using a technique referred to as fluorescence lifetime imaging microscopy, or NAD of NADH itself. And so fluorescence lifetime is a very specific technique that actually measures the amount of time that a fluorophore stays in its excited state after excitation. And we can actually measure this and see changes. And in the context of NADH, when NADH is free in the cytoplasm, it has a very short lifetime, but when it's bound to enzyme, it has a significantly different fluorescence lifetime. So this gives us a metabolic contrast of agent. By using fluorescence lifetime, we can actually get more information about the cell's metabolism at that, instead of just looking at intensity. And so the way that we use this is we use a technique called time-correlated single photon counting. And this is a really basic explanation, but if we imagine that we just centered our laser on a single point, we take advantage of our multi-photon titanium sapphire laser that has pulses at very distinct sequences at 80 megahertz. And these pulses are, because they're 80 megahertz, they're separated by 12.5 nanoseconds. And so what we can do is if we start our clock after the excitation, so we know exactly when we excite our sample, we can then start a time clock to when that photon from the emission spec comes and hits on our detector. And this gives us different time frames. And so if we hold that spot there and we continue to collect these different time frames, we can actually create a histogram at that spot that will then be able to mathematically fit in order to measure the lifetime at that particular spot. Now, if we do this at different pixels and create an image, what we create is now an image that no longer is looking at intensity, but is actually looking at the fluorescence lifetime decay of the entire field. So this would be the intensity of the image. This image is now giving us local areas of changes of NADH fluorescence lifetime. So what we did is we applied this technique to our in vivo area, and we created, this would be a fluorescence lifetime image of this exact area underneath the mammary imaging window. And what we found was by gating out just the tumor alone and minusing the bright FAD cells, and then gating the FAD high cells, what we found was that not only are we seeing these changes in FAD and NADH, but we're also seeing a significant decrease in their tau means, or their lifetimes, suggesting that not only are the FAD high cells changing in their intensity, but they are significantly different NADH metabolism characteristics. So we wanted to ask the question if this is correlative to our macrophage question. And so we crossed our tumor model with an animal model that has GFP that's expressed in monocytes. So this would include macrophages and neutrophils. And we did the same gating that we did before, where we looked at, like a cookie cutter, just the lifetime info of the bright GFP cells and compared that to the tumor itself. And what we found was a very similar trend. So the tumor cells that were bright in NADH had a significantly longer lifetime than the bright FAD cells, which was the same as the tumor cells had a significantly longer lifetime than the bright GFP monocytes. So this suggests that fluorescence lifetime is also giving us a similar contrast of these two different cell types. So what we also are interested in doing in the future is studying how collagen interacts with the tumor and how that can influence the local metabolism of the tumor itself. And so this is an image looking at just the brightest pixels of NADH, and what we can appreciate here is the heterogeneity inside of the tumor itself, of the lifetime of the tumor. So we have longer lifetimes and shorter lifetimes. They're not homogeneous. But if you look, it seems like in areas that are closer to the collagen fibers themselves, we can see a significant difference, relative changes between the metabolism closer to the fibers as opposed to far away in the tumor itself. And so in conclusion from this talk, I hope that what you got away from this webinar was our lab's work looking at tumor-associated collagen signatures and that they're prognostic markers for breast cancer research. That NADH, FAD, and second harmonic generation can provide the endogenous stromal and tumor cell contrast and provide us with a non-invasive way of imaging the tumor stroma micro architecture, and that you can use NADH film in vivo, and that this can provide in situ metabolic signatures in the breast tumor microenvironment. And what I also hope is that the data that I showed you today that was published in my paper was that FAD high cells seem to be high are CD45, CD68, and F480 positive, which suggests that these intensity changes are actually showing us a signature that's specific to stromal macrophages in the breast tumor microenvironment. So with that, I'd like to thank my lab, especially Patty, for all of her help in our work. I'd also like to thank our collaborators and Kevin Elisari for all his help and guidance in this research. I'd also like to thank funding and our collaborators at our different institutions, John Candelas and Julio and James Castercane. They've been instrumental in this project. And with that, I'd like to entertain any questions. Thank you.
Yeah, thanks very much, Joe. That's a great talk. Really interesting material there. So we will now open up the session to questions. As I said before, if you have a question, type it into the question section on your interface that you have here for the webinar, and we can go through them. A couple of things we have here already. One question was for the studies that you were doing with the window technique that you developed, what's the thickness of those tumors?
The tumors themselves can range in size based off of how advanced and how large it grows. We usually, at the larger end, they are as thick as 1 centimeter, and at the smaller end, they're probably closer to 500 microns. But we don't get that penetration depth when we optically image it. So we're just limited to just the area that's closer to the window itself.
Okay. So what is your imaging depth then when you're using a multi-photon technique here?
Yeah, so it's very scattering. So tumor tissue is really dense. So we go through a superficial area that's right below the window, and then before we get to the tumor. And I think that we really top out at about 200 microns of penetration inside of the tumor itself.
Okay. So have you obviously showed some interesting architectural features, especially with the collagen formation, the way it works, and then some interesting metabolic changes. Have you done any kind of control experiments looking at, removing the tumors from the animal and doing like cross-sections of the cross-sectioning of the tumors to verify that the kind of changes you're seeing that relatively close to the surface, when a couple hundred microns, is similar all the way through the tumor? Or
yeah, so our model, the polyoma model, is a multifocal. So what we actually see is many different tiny tumors that are kind of coalescing and behaving like one big tumor, if you will. And so yeah, we absolutely see all of our research that I presented first was actually all done by removing the tumor and slicing it and looking at their second harmonic generation. But if you're asking, did I do these control experiments looking at the endogenous fluorescence that I was describing with NADH and FAD and their lifetimes? It's interesting. I actually don't see a difference once I sacrifice the animal and fix the tissue. It seems to be something that is an in vivo specific event. So when I do try to do the controls to compare and contrast the endogenous fluorescence intensities, we really lose that contrast. They all look very similar, which is a really interesting technique. Interesting finding. Yeah.
Oh, it implies that the tissue has to be metabolically active in order for you to see these differences in the NAD.
Absolutely.
And okay.
The cells have to be alive.
Yeah. So do you have to? I mean, have you ever? How long will a tissue remain metabolically active to be able to show these changes, do you think?
That's a good question. So there's a lot of work where people in groups on campus here and all over in the cancer research field, where they remove the tumor and they put it into a three-dimensional environment in vitro. They call it a tumor spheroid, and they're able to keep it alive. But I don't I don't know. I've never studied personally the changes that you see metabolically from after you take it out and trying to keep it alive. And it seems to be a really rapidly degrading event. So if I, for instance, try to flash freeze the tumor right after I remove it and hope that I continue, like I freeze the metabolic features in time, it seems like the freezing or the fixation process alone removes that contrast.
Okay. So another question is because it's more related to the, I guess the lifetime imaging technology itself. So the lifetime images you showed, what was the size of the field of view? And the second part of that question is, how long does it take to generate, what's the scan time to generate that image?
Yeah, that's a good question. So we use it, I'm using a 20x objective. So the field of view is about 550 microns by 550 microns. And so about a half a millimeter in each direction. And the time frame that we did for these studies is each field of view, we let it scan for 2 minutes in order to collect the amount of photons that will allow us to have our fits with confidence.
Okay. Another question. So you showed a lot of data showing differences in architecture, especially related with the collagen fibers at different stages through the progression of the disease. Have you done any studies where you look at treatments to see if we can reverse those architectural changes?
That's a good question. No, I don't know of a study that specifically looks at reversing those changes. And I think it gets to a really good question there, Jeff, which is what is the cause of these changes, which is yet to be elucidated. There's many players that are involved in the tumor microenvironment, and what you could imagine is that perhaps what's realigning these fibers into that advanced tumor TAX3 would be cells bulldozing itself out. So cancer cells bulldozing itself out of the tumor stroma boundary itself. But there's also other cells that are inside of the stroma that are involved in depositing and creating new matrix, like fibroblasts that could be creating, and cancer-associated fibroblasts that could be creating a new template. And so that is something that we don't know yet. So we wouldn't really have a way of targeting the TAX3 signature until we can identify who's the major player involved in that.
Okay. Another sort of technical question related to the software that you had mentioned had been developed for you to help you analyze the fibers.
Mm-hmm.
Your colleagues is that specific for this particular type of analysis where you're looking at the collagen fibers, or can this be applied to any type of image where you've got basically appropriate contrast in the image and the structure is some sort of a fibrous kind of structure?
That's a great question. So I can speak to the point that it was written specifically for that context, but I can imagine that it's probably very relatively versatile. But that's probably more of a question for the developers themselves. I do know that people use it for different applications than just breast cancer, but I don't know if they have used it in other features beyond second harmonic and collagen. But I'm sure that'd be a good question for loci if you contact them.
Okay. Right. The last question I see here is, so were any of these studies you did longitudinal studies where you were repeatedly imaging the animals over different days, or were they all sort of single shot kind of experiments?
You the same animal over time?
Yeah.
I did. Yeah, you can do that. And that's one of the powers of what we're using for the mammary imaging window. But none of the data that I showed you today was actually looking at the same animal over time. We were doing kind of one shots at each data point. But that is something that you can do. Yeah.
Okay. All right. Okay. Well, there's no other questions. I'd like to thank Joe for his time and the great presentation. Really interesting material, Joe. If anybody wants to review the webinar, it'll be posted on our website by tomorrow sometime. And also, if you could take a minute as you sign out, we'd really appreciate it. If you could fill out the brief survey that'll be on your screen. And thanks again, Joe. It was a great webinar.
Thank you. Thanks for the opportunity.
Okay. Thanks everybody for attending.